Hello, Mr. B here. This is a little slideshow on forensics. Just going to cover a couple things: careers in forensics, fingerprinting, and then also hair. What you can learn from hair. So first of all, forensic scientists versus engineers. Well, forensics, that's the use of scientific technology in crime scenes. And the scientist, the forensic scientist, think about some of the shows you see on TV. That's the person that's going to be in the lab. And so they're doing more of the, I don't want to call it mundane, but they're doing the work that really requires some detailed following clear procedures. Why? Well, that's what you use in court, right? So they're going to be using their evidence that they find in the lab in court. So if if they're not precise and accurate, that evidence could be thrown out, even if a person was actually the person that committed the crime. So some of the things like you've seen on TV, it could be what a forensic scientist could be looking at is biological, chemical, or physical samples. And this is just a humongous list of skills. And a lot of these you could say is required of engineers as well. Some of the key things, patience. A lot of times you don't get your answer right away. You actually have to work pretty hard to get to your result. Meticulous and patience seem to work in hand. If you're not patient, you're not going to be meticulous. Obviously, one of the things, high degree of personal integrity. In other words, that the person is really going to make sure that they're doing the ethically and morally correct thing in their job, because in the end, someone could go to jail for life for a crime that they didn't commit if they messed up. So, I'm not going to read all these things to you. You can read that. The duties that a forensic scientist performs. These are some of the lab skills. They'll use some things called gas chromatography, and there's also liquid chromatography. We did one type of chromatography, the HIC chromatography with protein purification, but there's lots of different types of chromatography. They might use spectroscopy, which is basically a different way of looking at, it's like a fancier microscope, and they have infrared, called IR, and there's a UV and fluorescent spectroscopy. And there's microscopy. Again, basically these are just different imaging techniques. You could even say photography really is an imaging technique. There's lots of fancy ones. Those that go on to college, you'll see, you'll probably, if you do anything with chemistry or even biology for that matter, chances are you'll get exposed maybe briefly to some of these advanced imaging techniques, and certainly if you're a graduate student, that would be something that you would do on a regular basis. So a forensic scientist ultimately is going to be preparing a report for court, or it could be for the police, whatever. And some of those forensic scientists also will present in court. And obviously, just like you can see on TV, they can be subjected to some pretty intense cross-examination. Okay, so there is actually, you can go to school to be a forensic scientist. There's a 4-year degree, and this was just like, on average, that they'll have internships, and this gives you a feel of what kind of coursework you'd be doing a lot of, okay? Then there's areas of specialization. Some of these are pretty cool. Toxicology, someone that specializes in poisons, like, finding that the person died because of, some random chemical that maybe somebody that's not trained in that would overlook. Then there's people that can specialize in trace evidence. Entomology, and this is a study of insects. And you say, well, insects, why is that of importance? Well, within a matter of hours of a person dying, there's certain bugs that will come in and feed on you, and so depending on how long you've been dead, there'll be different bugs eating the corpse at that point. And so they can trace back to, okay, when did this person actually die by looking at what insects are feeding on the dead body. It's kind of morbid, but at the same time kind of cool. Um, and then there's a crime scene investigation, CSI stuff, and then death investigation. So a little more in-depth investigation. What that all means, to be honest with you, I don't remember. Forensics engineering. Now, this is different than forensic scientists. And here's the big difference. They're trying to answer why a product failed or killed someone. And, for example, why did the vehicle roll over? Could have accident been prevented? Forensics engineers look at airplane crashes, kind of morbid, but it is what it is. Buildings that collapse, looking for defects. And I have personal experience working with people that basically were called into court essentially as a forensics engineer. Yeah, they weren't a forensics engineer by training, but they got into that kind of as a matter of circumstance, and so called in often on insurance cases. So I have witnessed that. And with forensics engineering, a lot of them are going to have a BS, but they also might have a master's or a PhD. And in the case of the person that I work with, they did have a PE, which is basically they took a test to get a license to say that they're a professional engineer. Doesn't really make someone smarter, because I've worked with PEs that you wonder how they made it to work. It just means that they passed the test basically. But it does at that point in time exhibit that the person had some competency. Right. And then blood spatter. We've gone through blood spatter. I'm just going to let you know the type of things that would likely come up on the final. What a single drop pattern might look like, 6 inches, a foot. Basically maybe they give you like different single drop patterns and saying, oh, which one would have been the closest or furthest away, something like that. A multiple blood drip pattern where you get more splashing. The walking drip pattern, in other words, if you had a cut and you were walking, obviously the distance between drips is going to be a lot larger than if you were, or say it would be smaller than if you were running, for example. Okay, so if you're walking versus running, the drip patterns would be different. Vertical and horizontal arterial spurts, how they would look differently, and then impact angle blood stains. All of those we worked last class, which was pretty cool, and we'll be looking at that next class, and then going into the fingerprints, which is where I'm headed right now. So fingerprinting 101, this visual, a lot of the stuff we don't really have to worry about. The one that I will point out is this thing here called a delta. Okay, that's something that we're going to be using in a second. A lot of this other stuff, fingerprinting is a science in and of itself. We're just going to scratch the surface here, so we don't have to worry about all this other stuff. Now there's a FBI website, which I already have up, and I'm going to, oops, and I have to, well, do it this way. This isn't, let's see, Alt Tab. Let's try it this way. Put the microphone down for a second, and I'll click on the site. So this is very easy to Google search this. The FBI has some different things that you can click on, and one in particular is this fingerprint identification overview. It's a PDF, and it's pretty interesting. It goes into more details than what we have to worry about. But it gives you a basic intro into just some basic background on fingerprinting, and they have some more detailed stuff in here. I'm going to come back to this visual in a second. All right. So the first thing I want to point out is, okay, we talk about our fingerprints and the oils on our fingers causing the fingerprints, and really the palms of your hands don't make oils. The palm of your hands sweat. Okay, so the residual, you have like amino acids and stuff that come out with the sweat glands. That would be on your this would be the epidermis, would be the top. The dermis you don't want to be looking at, because that means you cut yourself. But the epidermis, which really is just the dead skin cells. We may see a fingerprint because of basically, amino acids and stuff that came from the sweat glands, that would be on the palm of our hands. Well, a lot of times people talk about the oils that are on our hands. Well, if our hands don't physically make oils, where do they come from? Well, if you see here, what this is a hair, and associated with the hair shaft is this thing called a sebaceous gland. That's basically an oil-producing gland. And that the reason why you have that is that ultimately is, well, one of the reasons, I'm sure there's multiple, is to protect your skin from infections. So this oil gland would be anywhere that you have hair. Well, your face obviously is where you have a lot of these glands, and so a lot of times the oils that end up on the palms of your hands actually came from you touching your face. All right, so that's how when you leave a print, a lot of times that oil came, if it was oil that you're actually seeing on a print, that would be from your face. But they can also pick up the prints from what would be the amino acids that have been left from your sweat glands. Now there's a couple types of fingerprints. There's the latent, which is an accidental impression, and it could be visible or invisible at the time of impression. In other words, if the latent fingerprint was left, like say, you grabbed a doorknob, um, you didn't mean to leave the print. You could have just wiped your head, so you had some oil on your hands, and so a lot of times these latent prints are smudged and distorted, and they can be really tough to get reliable data. Because just think about it, if you're a criminal, you're not going to go and just let's make a nice fingerprint and touch something very carefully. Like I'm sure everyone here at some point has been fingerprinted and how they have to roll your finger and all that stuff. Pardon me. That doesn't happen on a crime scene, obviously. Then there's so those are latent. Okay, so you might be able to actually pick up that fingerprint. It also could be invisible. Um, but the visible ones, those are called patent prints, and those are ones that are coming from blood. It could be from ink. It could be dirt, something that physically like contaminated your hands and then transferred. So that would be a patent print. All right. So before I give you the fingerprint ID quiz, I wanted to go through. This is another site here which ties it comes over to this page here. So this was what I did is I just typed in loops, swirls, and arches. Those are the three things that you have to worry about with fingerprints. All right. So this is the website handresearch.com, and what I liked was this visual right here. And so what it's showing is the three types that we really have to worry about in biotech. So we're just scratching the surface. Our loops, swirls, and arches. Here's an arch, here's a loop, and here's a whirl. Now, what you'll notice with an arch is it kind of swoops up and comes back down, all right? What you'll notice with the loop, and they've kind of highlighted this in yellow, is it has one delta, which they also call tri-radius. So tri-radius or delta, those are the same thing. So a loop, so it kind of can, this one would be from the left, so this would be a left loop, and it has one delta. But a whirl, which is kind of circular, so it's more like circular than the loop. And what another thing with a whirl is, if you've got the full print, what you should see is two deltas or two tri-radiuses, okay? Or tri-radii, I guess would be the better way of saying that. And so what this site has also, which came from the FBI website, and I already opened it up, is this here. So it gives you a little more detail. The arch, very similar. Here's it's called a tented arch. And now you've got different types of loops. Sometimes they'll call this, I think, a right loop, and I believe that this one would be called the left loop, just indicating the direction. I may have that backwards. And then they call this one a double loop whirl. Obviously, that's just a straight up whirl here. There's your two deltas. Then they have this central pocket loop whirl. And I think what they're talking about with the, here is the the loop part, but then it also has this whorl coming over there, and you know, again, you see a delta associated with it. So there are combinations of this stuff. On the final, I'm guessing that, you know, it would be something along the lines of something that's not a mix. They're not going to get that crafty. They would probably use, you know, a whorl or an arch, or, you know, maybe just a whorl arch, like a loop, one of these two loops here. It might be, you know, have those four pictures and then just say which of the following is a whorl or something like that. All right. So let's go back to the PowerPoint. Okay, so now let's just see how well you did. All right, so what do we have here? Loop, whorl, or arch? Hopefully what you're saying is it's an arch. Here's a tented arch. All right, and what are these? Loops, whorls, or arches? What do you think? Well, I see a delta here. It looks more like a whorl to me. Looks like we've got another delta over there, so I'm thinking that looks like a whorl to me. And then what's this guy here? We've got one delta here. It's coming from the right. Okay, that would be a loop. All right, so one last thing that I didn't mention I was going to cover, but I'm going to cover on this is hair analysis, because hair analysis is obviously another component of forensics, and the last of the forensics tools besides DNA testing, which we'll be rolling into soon. Um, but hair analysis is definitely helpful on a crime scene. It's not a slam dunk, and we'll go into why. Um, but one of the things that you can get from hair analysis is what part of the hair. Let's see. I gotta read this. Sorry. Why is hair an excellent structure to study for identification purposes? Well, what do you think? Why is it excellent to study? Well, you can tell a species. That's one thing. If you're thinking, well, what could you do with it? Yeah, you could tell the species. Is it human? Is it you know, dog? Is it cat? Is it a mouse? You know, think about the things you might find on a crime scene. And then the other question here is, well, okay, there must be a difference from species to species, and sure there is, and that's what we'll get into here in a second. All right, so the basic structural composition is the very, very center they call the medulla. Then you've got the cortex, which is the area from the center, the medulla, to this area right in here. So right in there is the cuticle, and I'm sorry, the cortex. And then the cuticle is a very, very external part. All right. And the cuticle, so I'm just talking about this part right here. The cuticle is protective coating, and that's what you'll see is like, looks like scales. If you've ever seen your hair, what you're going to see under a microscope, kind of looks like scales. And what's interesting, the scales will point towards the tip of the hair. That I would say might be a question that might come on the final, just pointing out some of the things that, you know, because we're just giving you an overview, so I just want to point out some of the questions where, yeah, that's probably fair game for the final. Another one that would definitely be fair game for final is, could you use the cuticle and and a hair analysis in general to help identify a suspect and ultimately convict them? And the answer is, unless you have the hair follicle, you can't get a DNA sequence. DNA is really going to be much more of a slam dunk than just the hair itself. So no hair follicle. The hair might be helpful circumstantial evidence, but it's really not going to lock someone up in jail for committing a crime. All right. One thing you definitely can do with a cuticle is you can use that to identify a species. Now we'll go towards that next section there, the cortex. And what's interesting with the cortex is it has these cells, they align in a kind of what they call an array, and it goes parallel length of hair. I'll give you a visual of that in a second. And that's where the pigment comes from, okay? So that's why I did brown here, because, hey, I've got brown hair. Well, I theoretically have brown hair. It's turning gray, which I'm going to be honest with you, it kind of sucks, but it is what it is. Y'all get there someday. And this is, you know, I highlighted this because this is really what someone's looking for, or a big thing they're looking for in a hair analysis. the color, shape, and distribution of the granules can be very helpful in saying, oh, this is definitely suspect Y is hair. That's what was found on the scene. Okay, so they're looking at the cortex. And then the medulla, and that's a structure that runs through the center of the cortex. And here's a little picture of that. And you could have continuous, interrupted, or fragmented. All right. And so along the medulla, you can measure the diameter of the medulla relative to the diameter of the hair shaft, and they put some math to it, so they give it a fraction. So they'll look at this medullary index, and so if you end up in this range here, they're going to say, oh, that's definitely human hair. And if we're in this range, well, then we're more, it's probably an animal. Okay, so again, you got to think about it at a crime scene. They're going to find all types of hairs. And here's some different species. The humans has a little break. A dog looks something like this, deer, mouse, cat, and rabbit. And we're actually going to be looking at the bulk of these on the microscope in the lab. And as I mentioned before, you find hair at a crime scene, it's not really gonna help lock in on someone in a crime, or it's not gonna be much more useful than circumstantial evidence. Again, to compare hairs from a perpetrator to to what was found on the crime scene, they'd be looking at the scale structure, the medullary index, and the shape. And yeah, that's evidential value. I'm just reading this. I honestly I hadn't looked at this slide. This is the last slide, and I skipped over it when I was prepping here. Sorry about that. Lives with degree of probability associated with questioned hair in a particular individual. I guess really in the end it just comes down to this is going to be circumstantial evidence. I guess this is why I've mentioned this. 11% of all morphological hair matches. So we find hair on a scene, we find hair of perpetrators, and so basically let's just say 10% of the time that even though morphological hair matches, in other words, looking under a microscope, it matches, that they actually are from different people. And so again, so the question is, so what part of the hair would be best to really quantitatively ID a suspect? And again, this would be something that would be fair game on a final. What would they be looking for? If we can find the hair follicle, from there you can do DNA evidence. You don't have a whole lot of DNA on the hair follicle, but you have enough. You have to mass produce it in a thing called the PCR, which we're going to be doing in the lab. Only we won't do your hair follicle, which we could. It's just a little bit harder. We're going to be mass producing cheek cell of yours in a PCR and then running a gel electrophoresis on that. Alright, so that's all I have to say, and we're all set with forensics besides wrapping up our blood exercise, and then we'll do some fingerprinting in class. Alright, so bring your guided notes with you to class, print them out if you can, that'd be great, and again, I'll just check it and give you credit for watching the video, and then we'll be using those notes to help us in a fingerprinting lab. Alright, thank you very much. Have a good day.